Okay, so I said how the mutation evolved over, or how the populations evolved through mutation. And mutation is just a change in the nucleotide, the sequence of the DNA, but you can't predict what's going to happen or how it's going to happen. And most mutations only occur in the somatic cells, which are all cells that aren't the reproductive cells. But when a mutation occurs in these cells, once the organism dies off, then the mutation dies with it, and it doesn't reproduce into the offspring unless the mutation aligns with that is produced in a gamete. And that can be passed off through the offspring, but only a small fraction of these spread through populations. So it can produce into like the next child and then that child, but it's not going to like spread out through an entire population, most likely. Point mutations are mutations that can be changed as little as just a single gene. So you could just change one simple gene and it would be a whole different thing. And most are harmless, but some can significantly impact the phenotype, which is what the organism looks like. And an example of this would be sickle cell disease. The reason that these occur is because much of our DNA and our genomes do not code for the protein products. And if the gene with the point mutation codes for proteins, it may still have little effect because they do not alter the protein's amino acid composition. So that might not even change much. Mutations are usually very unlikely to improve the genome. On rare occasions, a mutant allele may actually make its carrier better suited to the environment, enhancing reproductive success. And this is more likely when an environment is changing and mutations that were once selected against become favorable. So like if the environment changes and the mutation adapts and allows it to change with the environment, it may become successful for the organism instead of hurtful, which is a positive. And mutations that alter gene number and sequence. Chromosomal mutations that delete, disrupt, or rearrange many loci at once are almost certain to be harmful. In rare occasions, chromosomal rearrangements may be beneficial for some of the same reasons. Gene duplication is an important source of variation. These variations, just like other chromosomal mutations, are nearly always harmful, but there's a rare chance that they aren't. Smaller pieces of the DNA are often introduced into a genome through the activity of transposable elements, which we learned about last chapter. If the duplicated segment does not have severe effects, then it can continue over generations, providing an expanded genome with new loci that may take on new functions by further mutations and subsequent selection. Okay, and new genes may also arise when coding portions of the genes are shuffled within the genome. So if you rearrange it and everything, you're going to get a new sequence than you would before. There have been many beneficial increases in gene numbers which appear to have a major role in evolution. Close ancestors of mammals carried a single gene for detecting odors that had been duplicated through the variety of mutational mechanisms, which now allow humans to have 100,000 or just 1,000 different olfactory receptor genes, but only about 60% of those genes are inactive. So even though this mutation has evolved over time, we still don't use all the new genes that we have, even though we have them to smell better. And mice have about 1,300 olfactory genes, and only about 20% are inactive, so they use more of their olfactory genes than we do that have mutated over time. Mutation rates tend to be low in animals and plants, averaging only about a single mutation every 100,000 genes per generation. So they're not very common, but microorganisms viruses with short lifespans have mutations that can rapidly generate genetic variation. So that's why every year there's new flu shots and all the other different types of vaccines, because the gene and the virus is always changing through mutations, and you need something to prevent it, because they will adapt to the old vaccine, which won't stop it anymore. A single drug treatment on viruses will probably never be effective, because mutant forms of the virus that are resistant to a particular drug are likely to rapidly multiply in relatively short time, which is kind of what I just told you. And double drug treatments rarely work for a long time, because viruses with double mutations can form a resistance against both drugs. So if you're using, at the beginning it'll probably work if you're using two different drugs to stop the one virus. One of them is bound to work, but soon the virus is going to form a resistance against both drugs.
Can you think of any examples?
Like the flu?
Well, okay, the flu. What about it?
Well, you have to get you have to get a flu shot each year, because the virus is constantly changing, that the flu shot you got last year isn't necessarily going to prevent you from getting the flu this year.
Okay. Now, what about other viruses like HIV?
Oh wait. Hold on. It might be on my.
I mean, if you're
Okay.
going to come to it later, that's fine.
Yeah, I'm not sure if I put it in here or not, but I
Okay.
can see.
All right, and then the other thing I was going to ask you, and like I said, if it's something you have later, that's fine. What about how do these viruses, which are non-living entities, evolve to become resistant to certain antiviral drugs and to vaccines even? Because it is true, you got to get a new, you're right, flu vaccine, you got to get a new one every year, because new strains of the same virus all the time. So how are they getting mutated?
Aren't they just like they changing their proteins, and when I don't know how to describe it. When
They're changing their proteins, they are changing their genetic sequences, but
yeah.
the question is, how are they doing this?
Yeah, like when the outside changes, then they change with it, but they can't do it without the host cell.
Right, that is true. They do need a host, and that's why the mechanism is a little different than what you see with other work with actual organisms. There's one process, it's called genetic reassortment. Are you familiar or no?
No.
Okay.
Okay. Sexual recombination is the process or act of exchanges of genes between chromosomes, resulting in different genetic combination, and ultimately to the formation of unique gametes with chromosomes that are different from those of the parents. This is far more important than mutation on a generation to generation time scale in producing the variations based on adaptation possible. Almost all phenotype generations based on genetic differences result from recombination shuffling of the existing alleles in the gene pool. So when you have the gene pool and they all shuffle around and rearrange, you're going to get different phenotypes than the parent has, and you're not necessarily going to have what the parent had. Populations contain countless possible mating combinations, and fertilization brings together the gametes of individuals that are likely to have different genetic backgrounds. So if you bring two parents together, they're not gonna have the same genetic like sequence, and then when you put them together, they're gonna form an offspring that doesn't have the same as them, and then when that offspring mates with someone else, theirs aren't going to be the same, and that's how it keeps changing over time, which is why it's more for generation to generation if you're trying to look at the difference between them. And then sexual reproduction rearranges alleles into fresh combinations every generation, which is what I just told you. Bacteria and many viruses can also undergo recombination, but they do so less regularly than animals and plants, and often in ways that allow them to cross species barriers. For example, the
Just
E. coli frequent culprit in food poisoning, which are actually mosaics in individuals composed of cells of two genetically different types, the genes from different types of bacteria. So they are two different types of genes put together, and that's what makes them a different type of bacteria, which is found in food poisoning. And the ability of the pathogens to evolve rapidly through such extensive recombination, combined with their high mutation rates, makes them especially dangerous to their enemies, because they change and then you have to fight them off again, and then they change and then you have to fight them off again, and so it's an ever-going